This is the continuation of the lecture on the introduction of nervous system. Here we are going to talk about the cells of the nervous system. When we are talking about the cells of the nervous system, there are two main types of cells that make up the nervous system. Those are neurons, or the excitable cells, and the supportive cells, which are non-excitable cells. Neurons are the main type of cells which form the nervous system. That is because they are the excitable cells which can receive stimuli, and the impulses are conducted through neurons as action potentials. These signals are transmitted to other neurons as well as the effector organs of the nervous system. The other type of cells is the supportive cells. These cells are non-excitable cells. They are called Neuroglia or glial cells. They are important for the support, nourishment, and also for the protections of neurons, as well as they perform some other functions. When we are considering the structure of a neuron, there are three main parts of a neuron, that is the cell body and two types of cellular projections. They are axons and dendrites. You can see them in the image below. The cell body of a neuron is called neuron cell body or soma. Most of these neuron cell bodies are located within central nervous system. The clusters of cell bodies that are located in central nervous system are called nuclei. When these clusters of cell bodies are located in peripheral nervous system are called ganglia. The rough endoplasmic reticulum located in neuron is called nasal bodies. One type of cellular projection is called dendrites, that is because it has a branching organization. And the other type is called axon because it has a straight alignment and also uniform diameter. These axons are also called as nerve fibers. Axons can vary in length from a few millimeters to more than 1 meter. Usually, an axon arises from a cone-shaped area of the neuron cell body. It is called axon hillock. The beginning of the axon is called the initial segment. The cytoplasm of an axon is called axoplasm. Its plasmic membrane is called axolemma. These axons finally terminate by branching to form small extensions. These small extensions become enlarged in order to form structures called presynaptic terminals. The synapse is the junction between two neurons. The neuron that transmits a signal towards the synapse is called presynaptic cell, and the cell that receives the signal is called the postsynaptic cell. The space between these two cells is called synaptic cleft. You can see the synaptic cleft and presynaptic cell and the postsynaptic cell in the image below. Within the presynaptic terminals, there are small, numerous membrane-bounded secretory vesicles which contain chemicals called neurotransmitters. The action potentials or the nerve impulses which conducted along the axon finally reach the presynaptic terminals and then stimulate the exocytosis of these neurotransmitters from their vesicles into the synapse. Then the neurotransmitters cross the synaptic cleft. Then they can bind to the membrane of postsynaptic cell. This binding leads to stimulation or inhibition of the action potential in postsynaptic cell. Let's see what are the different types of neurons. Neurons are classified according to either their function or their structure. The functional classification is based on the direction in which action potential is conducted. Those are sensory neurons, motor neurons, and interneurons. Sensory neurons are also called afferent neurons. They conduct action potentials towards the central nervous system. The motor neurons or efferent neurons conduct action potentials away from the central nervous system towards muscles or glands. Interneurons conduct action potentials from one neuron to another within central nervous system. When we are considering the structural classification scheme, that it is based on the arrangement of the processes that extend from the neuronal cell body. There are three major structural categories of neurons called multipolar, bipolar, and pseudo-unipolar. In multipolar neurons, they have many dendrites, but just a single axon. The dendrites vary in number and also in their degree of branching. Most of the neurons within the central nervous system and motor neurons are multipolar. When we are considering about the bipolar neurons, they have only two processes, that is, one dendrite and one axon. The bipolar neurons are usually located in some sensory organs, for example, in the retina of the eye and in nasal cavity. The other type is pseudo-unipolar neurons. These neurons have only a single process which extends from the cell body. But this process divides into two branches a short distance from the cell body. One branch extends to, say, central nervous system, while the other extends to the periphery and form dendrite-like sensory receptors. But these two branches function as a single axon. Most of the sensory neurons are pseudo-unipolar. This image gives you the idea the differences between the structure of different neurons. So now we are going to talk about the non-excitable cells of the nervous system. They are called Neuroglia. First, we are going to talk about the supportive cells of the central nervous system. They show different types of functions. The first function is that they participate in forming a barrier between the blood and the neurons. This barrier is called blood-brain barrier. And also they participate in phagocyting of foreign substances in order to protect the brain from foreign substances. And also they are involved in producing cerebrospinal fluid. They also form myelin sheaths around the axons. There are four main types of neuroglia in central nervous system. They are called astrocytes, ependymal cells, microglia, and oligodendrocytes. Each of them have unique structural and functional characteristics. Astrocytes are star-shaped cells. Its cytoplasmic processes extends from the cell body, and these extensions widen and spread out to form foot processes. These foot processes cover the surface of blood vessels, neurons, and the pia mater. Pia mater is a membrane which covers the brain. So in the picture you can see the astrocyte and how it forms these foot processes. In this way, astrocytes form a supportive framework for blood vessels and neurons of the brain, and it also helps to regulate the extracellular composition of brain fluid. This is a real picture of astrocytes. The ependymal cells line the cavities of the brain and also the central canal of the spinal cord, so you can see it in the image below. These specialized ependymal cells, together with blood vessels, form the choroid plexus. They are located within certain regions of the brain cavities. The choroid plexus are the organs which secrete the cerebrospinal fluid, which flows through the cavities of the brain as well as in the central canal of spinal cord. Some cells of the central nervous system become mobile and phagocytic in response to inflammation. They phagocyte necrotic cells, microorganisms, and other foreign substances that invade the central nervous system. So these type of cells are called microglia. You can see a real picture of microglia in the picture on the right hand side. The neuroglia called oligodendrocytes have cytoplasmic extensions that can surround axons of neurons. If these cytoplasmic extensions wrap many times around the axons, then they form a myelin sheath. A single oligodendrocyte can form myelin sheath around axons of more than 2 neurons. So, oligodendrocytes are the cells which form the myelin sheath in central nervous system. Now let's see about neuroglia of peripheral nervous system. The main two types of cells are Schwann cells and satellite cells. Schwann cells wrap around axons of neurons. If they wrap many times around an axon, it forms a myelin sheath. Each Schwann cell forms a myelin sheath around a portion of only one axon. The satellite cells surround the neuron cell bodies in sensory and autonomic ganglia. These cells are important to provide support as well as nutrition to the neuron cell bodies. These satellite cells are also important to protect neurons from heavy metal poisons such as lead and mercury. That is, by absorbing them and reducing their access to the neuronal cell bodies. These images give a summary on the neuroglia of the central nervous system and the peripheral nervous system. Please go through them carefully and get a good idea about these cells. So, this is the end of the second part of the introduction of central nervous system.